Hello, everyone. Good evening and a warm welcome to all our audience today who has joined us for this special webinar session. If you can hear me clearly and if you are ready to unlock the future of biology, give me a big yes in the chat box right now. Come on, let me see that energy scroll up. Wonderful. Yes, Anirban Kar, Sajil, good evening to all of you. Salil, thank you so much all for your responses. Fantastic. So that is the energy actually what we need today, right? All right. So folks, let's be real for a second. See, we have all these days thought biology to be just about labs, petri dishes, microscopes, right? But look around you. The world has changed. What we used to do 5, 10 years back, we don't do that anymore. Today, our curriculum in college, our experiments, our courses, they all look very different to you, right? So today, biology is giving us the questions, but data holds the answers, right? And bioinformatics, well, that's the bridge which connects both of it. Yes, very good evening to all the participants. Thank you so much for joining Pooja, Himadri, Sunita, Muzazira, Majbeen, Yatin. Yes. So I'll give you one more example to all of you. Imagine you have a library with 3 billion books. So this is roughly the size of a single human genome. Now try finding one spelling mistake in one book that causes a disease. Impossible for a human, right? But for a bioinformatician, it's just a piece of cake. So that is the power you are here to learn today. And here is why this matters specifically to you. We aren't just talking about science, we are talking about your career. Did you know that the global bioinformatics market it's set to smash past USD 25 billion by 2026 and that is massive. But here is the catch. While the funding is here, what we don't have are the trained professionals. So there is a huge shortage of people who speak both biology and code. Today we are going to fix that. We are going to turn you into that top 1% talent, right? And I also have a promise to you. If you stay with us until the very end, I have a special surprise gift for everyone attending live here. Mind it, this is not for the people who are going to watch the recording. It's only for the people who are going to attend us live until the end. So it's a thing. I'll not disclose the surprise, but it's something that is going to fast track your learning and it's completely free. It's a gift from Biotechnica, but only if you hang around for the big reveal at the end. So who's excited? Type I am ready in the chat box. Come on. Yes? Are you excited? Right? Bring it on. Right. So before we bring in the experts, I need to know who am I talking to. So, like, are you a student? Are you a researcher who is working in the lab, or you're just a career switcher looking for some upscaling, or just a working professional? So let me know in the chat box later, right? And so I think we have a great mix. We have a we have majority of students and researchers here as I assume. Yes. So okay, we have a lot of students here. Perfect. So this session is specially tailored for you, right? And also the professionals as well. Researchers are going to be interesting for you also, right? So before the wait is over, we have two absolute powerhouses of the industry with us today. So first up, we have with us today Dr. Munikumar Manne. I, uh, I, I would like your support to welcome our esteemed guest speaker for the day. Thank you so much, Doctor, for joining us today. And we are delighted to have you here and we look forward to your session. So Dr. Manikumar Mane, he's a PhD in bioinformatics and he's a director academics and trainings at Manna Biotech Private Limited, Hyderabad. And Dr. Munikumar is an accomplished bioinformatician and academic leader with expertise in genomics, nutrigenomics, neurodegeneration, and precision medicine. He has over 38 publications, multiple book chapters, and extensive experience in computational biology, AI-assisted drug discovery, and next-generation sequencing. Dr. Mani has guided more than 100 under postgraduate dissertations, served on editorial boards, and led national and international research programs in multiomics, molecular docking, and therapeutic design. Dr. Mani's work bridges advanced bioinformatics with real world applications in healthcare, pharma, and biotechnology. So, audience, you cannot miss this for anything in the world, right? So, take your notebooks and be ready for Sir's session. Thank you once again for joining us here, Sir. Our next speaker for the day is Dr. Neeraj Kumar. You already know him. He doesn't need any introduction. Dr. Neeraj Kumar is a computational biologist who uses artificial intelligence to speed up drug discovery, and he is also a bioinformatician. Yeah, Dr. Kumar has earned his PhD in bioinformatics from CSIR IHBT and ACSIR, then completed postdoctoral research at Pennsylvania State University, where he built algorithms for antibody analysis. He's skilled in tools like TensorFlow, PyTorch, RDKit, GROMACS. He has published in leading journals such as Journal of Cheminformatics and Medicinal Research Reviews. And he has also cleared several competitive exams like UGC NET, GPAT, NIPER JEE, and GATE also. So, Sir is going to walk you through what career roles exist in this field, where they work, what will be your learning roadmap, and and obviously it's going to be a game changer. Those who are looking to excel in this field, right? Yes, so Salim says, cool. Yes, I love support from Biotechnica. Thank you so much, everyone, for your support and enthusiasm. So, without much further delay, let's move on with our session. Dr. Muni Kumar, I would request you to kindly take over the controls and start with your session, please.
Yeah, thank you, Urmila. Hi, everyone. Very good evening. So let's share my screen first. Yeah, yeah. Urmila, could you confirm that my screen is great now?
It's visible, sir.
Yeah, fantastic. Actually, the Urmila is introduced myself. It's a Dr. Muni Kumar. Okay, I'm complete. There is a bioinformatician. And thank you very much for the biotech for inviting us. There's a guest lecture on how the career development, in especially the bioinformatics. See, as a biologist, they have some fear about how to handle this fellow, how to handle this fellow. So the people having a lot of fear how to handle the computer, how to handle the program. So if you're able to handle the test tube, yes, you are expert in this point of. But while coming to that, the biologist handling with a computer, handling with there is a patients are it's a little bit fearness and all. But don't worry, I promise you that. So today's section will be clear your fearness and followed by how to handle your the programming. So before we just start, so bioinformatics very well known subject, but the most of the criteria. But by coming to the introductory program, like so it is dealing with there is a biology. Of course, as a biologist, it's dealing, of course, the possibility. Then by coming to the mathematics, so there always be confusion like mathematics with biology is two different subjects and all. And there is another guy, there is a computer application, the third person. So that means to say is that bioinformatics can be able to handle the computer, is able to handle the mathematics and able to handle the biology? Yes, of course. Yes, of course. But the point of understanding, sir, I want to be grow in the bioinformatics field because of my career having the microbiology and biochemistry, or even go for any other biology subject and all. So my opinions always be like, see, don't jump to the bioinformatics and understanding the bioinformatics. That is very important. So keep it your the biology. For example, if you take the microbiologist, so how you are going to understand the bacteria and how you are going to understand the virus, how you are going to understand there is a fungal infection and all. So in that criteria, how you are going to implement the bioinformatics application. That is important. So that means to say that you are extending your basics to in advanced with respect to that there is a career with respect to in the industry. That is playing a major role. So what bioinformatics can do? So bioinformatics can do, there's a lot of, it's like that is I mean per movie based. Okay. So what bioinformatics can we do that? See, the bioinformatics can do, there is a protein analysis, there is a genomic analysis, proteomic analysis, there is a database development and database management, transcriptomic approach, and you can go further the pattern recognitions, clustering, classification, whatnot is a lot of things and all. But the question is, can I adopt? Can I adopt this syllabus to the my subject? That is very important. How much is useful for? So bioinformatics is look like an ocean. How you are look like a microbiologist is the ocean. The same thing criteria for the bioinformatics also is the ocean. But how you are going to adapt that ocean, that is important. Of course, we have a lot of mind-blowing activity here. We have so a lot of criteria to be there is a we are going to be discussed about some case studies and the very basic of central dogma. So if you know the central dogma very perfectly, yes, you are the expert in the biotechnology with respect to there is a biology student, and you are understanding how you are going to implement in the next criteria. And we are going to be touch some of the databases and all, and we have a knowledge about that molecular docking, how we are going to do the docking and dynamic simulation, and very recent topic at the next generation sequencing. And very boom topic is the cell signaling, like especially the single cell, how is going to be sequenced in the process. Of course, the agenda is another one is that how you are going to be enhance your industry skills with respect to the drug discovery and computer drug designing and some of the AlphaFold activities, some AlphaFold, there is a case studies and all. And what are the technical tasks we require? And what are the technical in the sense like how is the programming required? And how we're going to be grow in the career strategy. Okay. Sir, we are already fed up with a lot of subjects and all. We have a microbiology, we have a biochemistry, we have a biotechnology, a lot. Sir, you are explaining about the Python, you are going to be explaining about the R, how you are going to implement. So that's why I given the introductory in that. How you are going to be implement, how you are going to be extending your basic science to the, especially in the bioinformatics. That is very important. Okay, and what about the perspective, especially in the history-based bioinformatics? See, there is a lot of eras are available. So the first era it is like there is a sequencing based, like there is Dell, who is the mother of bioinformatics, and the later it is changed to as of now it's integrating with the multi-omics. The omics is very simple, like genomics, proteomics, metabolomics, and pangenomics. So these are all the multi-omics we are going to be adapting in the different sequencing based. And followed by another era it is started in the data wise, like there is a AI driven, especially how we are going to be artificial intelligence can be interacted with. Sir, we are already there is a lot of subjects related to that biology, microbiology, and all. How we are going to be understand the AI driven information and how we're going to be add in our subjects like there is AI driven research and development criteria. So don't worry, today we are going to be crack that one, especially the AI based now. Then what market is required? So what market is especially the industry is required. So if you understanding the NGS, especially next generation sequencing, with integrated with the AI, artificial intelligence, especially, and you are making the program in the cloud computing days. So actually the career right now industries are looking. And one more thing is that there is a precision medicine, especially the personalized medicine. This is the evergreen subject. Until and unless life, our precision has medicine has to be grow in the next stage. And I told you that the central dogma. If you understand the replication, of course, the replication is there is a copy of the DNA. And the transcription DNA can be transferred to the RNA. That is there is a transcription process. The transcription after that there is translation by the proteins and amino acids. If you understand this concept, your bioinformatics is easy. There is no other than this one. There is no other than if you take what not programming, what not the scripting, and what not in the AI, artificial intelligence, what not in NGS, everything is under in the central dogma. So make sure people has to be understand, guys. You should be understand the what is the basics. Without basics, there is no application point. Fine. And I just explained the genomics. So the genomics is nothing but the total content of the gene, blah 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 blah. The point of understanding like how you are going to be genomics can be integrated with NGS, how we are going to integrate the whole sequencing, how we are going to integrate it to a whole exome sequencing, how we are going to integrate the pangenomic approach for the pathogens activity. That is in the genomic activity. So if you go for the transcriptomics means it's very simple how you're going to be expressing the gene. And either is regulated, either is down regulated. And with respect to that, which is the metabolic pathways, that is the transcriptomic approach. And proteomic approach, of course, it's a protein-protein interaction. If you disturb one protein, if you disturb one gene, if you disturb in one network, how your going to be protein can be interacted with the other gene. That is something about the proteomic approach. And we have certain database. You may ask the question like, sir, why we require a database? So the initial class we have explained about that we have a lot of library books. We have a lot of library with respect to lot of databases. And with respect to DNA, RNA, protein structure, metabolism, a lot of. But how you're going to integrate with the programming, that is required. If you understand the primary database, like how you're going to be download from nucleotide sequence, the GenBank, how you download protein sequence from the genome, how you download protein structure PDB. This is the basic understand. The secondary database we can implement. There is the input informations too. Especially we can download the three-dimensional structure, especially by the PDB protein data bank. And how you're going to visualize in the different applications. That is very important strategy in biological databases we'll learn. And so this is the customized databases. So if you are a biologist, the maximum, the basic understanding, like for example, if you would check there is a bibliography data or literature data, we can retrieve from the PubMed. Sir, I want to go for the genomic database. Like I would retrieve from Enrich, especially the NCBI, National Center for the Biotechnology Information. Yes, I can retrieve. If I want to go for any disease oriented or metabolic based, or if you want to go for the enzyme based, or I want to download, I want to retrieve some chemical compound. So this is the customized database, especially who the jump to the basic students. Like there is a basic understanding, the basic understanding of bioinformatics. So make sure this is will you give you the complete picture of what we'll do with the bioinformatics. Right. And there is another objective, it is like computer drug designing. This is the evergreen subject. So still until Allah life, so the drug designing has to be there. You know very well about that one crisis, one pandemic situation at the COVID-19. We might be seeing a lot of drugs like paracetamol, remdesivir, hydrochloroquine, a lot of drugs are available. But as of now, there's now, there is no drug for COVID-19 because of the still research is going on, a lot of that. And if you understanding the drug designing, especially structure-based drug design, it's very simple. If a structure is available, three-dimensional structure is available, yes, we are going to be taking out the structure-based drug designing. And another alternative is a ligand-based drug designing, like QSR, quantitative structure activity relationship, and the pharmacophore modeling, similarity search, virtual screening. This is all stuff is available in the ligand-based drug design. And there is another scenario, just like to understand the basics, like if you available structure and the ligand, we can go for the structure specific designing. So if you're not available, there is a protein, you can go for the homology modeling, and you can go for the there is a ligand based design. If it doesn't have any protein and doesn't have any ligand, please start our activity by reading the more article how to do and all. And there is another objective, guys, the sequence analysis. Sequence analysis is the basic step in bioinformatics, basic step in the biology, especially you want to run any programming language. If you understand there is a local alignment, the global alignment, there are a lot of alignments are available. If you understand sequence alignment, I'm sure about you are the expert in the bioinformatics. You are the expert in the bioinformatics. So if you want to know that there is a sequence alignment, you are using the BLAST, blah blah programs and all. So the basic of this, if you understand the basic analysis, especially sequence based, Like I want to go for the family, which family belongs to the my protein and all. I want to construct the phylogenetic tree. I want to go for the database search for especially the BLAST, basic local alignment search tools. These are all applications are available. These are all applications if you understand in the sequence analysis. Yes, you are the pro in bioinformatics. Fine. And I just one objective that is the SBI. So don't call me that is a State Bank of India. It is just it's a structural bioinformatics. The structural bioinformatics it's a one objective to understand, especially the molecular docking analysis, molecular dynamics analysis, and especially that is a structure-based drug design. So that's what. If you know the structure, yes, you know the function. If you know the structure, definitely you can know the function. So how to download from the structure? Yes, we know very well about that is a protein data bank. And we know what is the biological function is available, especially structure based, and how we are going to implement in the drug designing process. So this is my evergreen question when I'm studying from MSc and PhD and I have experience and all. So question is very simple. The question is very simple point of understanding. Like, see, we are able to sequence, we are able to sequence, we are able to structure, but why we are not going to start treatment very faster? So if you take the microbiology, if you take the microbiology means, sir, I want to be grow the selective media, I want to go for the selective bacteria, then I will go with the selective treatment based. If you go for the virus, also same thing like. If you go for the biochemistry in the pathology means pathogenesis process or the metabolic process, which metabolic pathways is influenced. But in we are in the AI era. We are in the AI era. This question I got an answer after 10 years because of the NGS is fulfilled the maximum activity and the AI is also it's occupied with the there is a sequence and structural level the treatment it is started. You may ask the question what is the successful story for there is any the sequence and structure. Yes, the COVID-19 is a successful story because of the successful story. See, if you want to go for any sequence level or if you want to go for the treatment level, the COVID-19 within 1 year or 2 years, we got a lot of vaccines because of the structure is available, sequence is available, and integrated with the AI. The people are right now, there's a lot of vaccine. Of course, the side effects are secondary criteria, but still, if you have it, means again the third wave, fourth wave is coming something around means fourth wave or fifth wave. Yes, we are ready to do for that. Then I want to go for some case studies with respect to that. This is the one of the paper published in Biomolecules and DPI journal, which is having the six impact factor journal. And this is the FATP guy. He's the guy, it's like there is an overexpression and followed by the deficiency. If the overexpression deficiency, it can change the cancer condition, especially in the breast cancer. So what is the function of this guy? Is like he has to be transferred fatty acid biosynthesis. It has to be transfer the fatty acid transported inside the cell and out of the cell. But we taken a challenge here. We utilize there is a AI integrated homology modeling. You might be aware about that there is the AlphaFold. You might be aware about AlphaFold. Recently they got a Nobel Prize for the AlphaFold. Yeah. So we utilize the AlphaFold to construct the homology model, especially the FATP one. And we perform there is a validation process. Guys, it's very simple. When you are doing any experiment, when you are doing any analysis, when you are doing any programming, the validation is playing a major role. What you've done, what you are expressed, what you are analyzed, anything to be, it has to be validated. The validation having the next role, next criteria, next strategy to either you've done the perfect work, the perfect analysis, perfect examples. In that point of view, validation is very, very important. So we are validated by a lot of, there's a Ramachandran plot, ProQ, PROSA, and a lot of criteria are observed. And this is another, I told you that there is a COVID-19, which is one of the, there is the AYUSH CCRS group from, there is a, especially in India. So we have a collaboration with the I-64, and this is the guy, especially from COVID-19 M protease. We perform the molecular docking. We perform the molecular docking and dynamics application with respect to the Schrodinger and with respect to the discovery studio and the two analysis we compared. And there is a one molecule which is interacted with the M protease of COVID-19. And we are validated by the Ramachan. There is a root mean square deviation and root mean square fluctuations by. And another one is that there is a MMGB assay. So this is the techniques we can use for the molecular dynamics applications. And there is another point. So I told you there is a validation is very important. So how you are going to validate this criteria? Yes, we are validated. We are validated, and it is made there is a policy standpoint, especially for there is a CCRSA group. So the paper is published in Journal of Ayurveda Integrated Medicine, and the people are a lot of. And when you are giving that there is a I-64, especially the mild and moderate patient for the COVID-19, they are treated. They are treated, especially the COVID-19, especially by the I-64 compounds. And this is another guy. This is a very fantastic story. This is a fantastic story. I think so. Maybe most of the people might be aware about these leaves. So this is the Tinospora caudiflower leaves, so which is available in normal in India, especially in Hyderabad or Bangalore or anything. So we in wherever big bush is available, yes, our Tinospora caudiflower is available. So the question is, everybody thinking about the pathogenic process. Everybody thinking about that. There is a especially where is a pathogen or microorganism enter into the body or virus is enter into the body. My question is very simple. See, you are not going to stop that there is an entry of bacteria or entry of virus because of that is not our hand, but how you are going to be treated. So the first question is, I'm not going to treat with the patient first. I don't go treat with any pathogenic process, but I want to be make sure that I want to make immune system, how you are going to enhance the immune system. That is very important. So in this point of view, we check the crude extract the genus Paracordiofolia, and we check with that, especially diabetic patient, especially there is a low immune system people, and there is a TGF. TGF means is the transforming growth factors beta, and especially another the tumor necrosis factor alpha, and both the protein having the very good information, the very good interactions with, especially cardiofoliocyte. This is the compound which is available only in the Tinospora cardiofolia. And the rest of the compounds may be available in the another scenario on the plants, like there is a phytochemicals or flavonoids are available. But the question is how you are going to be molecular interactions with, especially the Tinospora cardiofolia. And this is another one is that molecular dynamics. I think so maybe everybody aware about, but the basics people, the dynamics will give you the enormous information. There is a huge GB line. How your going to protein is interacting with the compound, having the fluctuation site, having the more it's having the more deviation, or it's having the close relevance and all. Yeah. And of course, the validation, I told you the validation has to be done. There is a root mean square deviation and root mean square fluctuation. So what is meant by root mean square deviation means? If you are docked, if you are put with the compound, how you are going to stabilize the particular period, the particular time, the particular seconds. So we are giving 10 to the power of minus 9 second of fluctuations. So it means we can call as a nanosecond. So in that 100 nanosecond period, how the protein is holding your drug, how your protein is holding your drug molecule or any natural compound, either having the interactions good or not. That will be studied by RMSD, and RMS root mean square fluctuation, then the radius of gyration and solvent oxide surface area. But of course, it is required if you go for sir, I want to run the molecular simulation studies, but my laptop is not sufficient. There is a lot of programs available in nowadays because of, we have to say thanks to the AI especially. So because of if you take there is a Google Colab. The Google Lab is an open source for the student, open source for that for the people who are having limited resources. And there is another source is the Kaggle. The Kaggle is another source. It's giving there is a GPU based, graphical processing unit based. So if you want to be write any program like the Python based or the R based, the sources are available. Makes sense, yeah. And I told you, if you disturb any one protein, if you disturb any one protein, this is the FATP1, this is also known as FATP1. If you disturb the protein, what is the rest of the protein having the changes? Having any changes? The each line indicates that there is a cellular level and biological level and a metabolic level. And what if you disturb any protein or any gene with respect to that other genes or not? Yeah, so we have a lot of database like this, string database. David, I'm so sorry, it's not a David, is not a person. David is a database for annotation, visualization, and integrating discovery. And there is another guy's name is the Shinigo. Shinigo right now is a competitor for the David analysis, and the Shinigo is having a lot of information related to that, how the protein is interacted with other protein, having any metabolic wise, any cellular wise, any even the chromosome positions also. Yeah. And another technique, and the present the boom topic is a next generation sequencing. So the previous days in the world means last 10 years back, if you go for the NGS, it's a cost effective. It's a very cost effective, like if you go for the big project, only the people used to go for NGS, especially the transcriptomic and whole genome sequencing, whole exome sequencing. But nowadays are in down. And there is another scenario, it is like there is short sequences and there is long read sequences. Okay. And we have from monobiotic, especially the long read sequencing, especially the nanopore sequencing in the back bio. And this is a very smart guy, this is a very smart guy, and it is not more than our mobile. We can sequence at a time 96 samples at a time. We may get within a 2 to 3 days, we may get the whole transcriptomic sequencing, or whole exon sequencing, or whole single sequencing activity. So the cost effective or came down. So it's not in the millions right now, it's coming to the dollars base. Yeah. And if you go over the pangenomics, the people are having a lot of sorry, um, movies like there is a pan movies and the global movies, something around. The question is why it's a pangenomic approach is not there. See, the pangenomic approach is nothing but which is the gene influencing, which is the gene is influencing. Sorry, yeah, I'm so sorry. See, the pangenomic approach is a very simple criteria, which is the gene is influencing the maximum cancer. If you take one protein name, there is a TP53, tumor necrosis factor, or tumor. Yeah, TP53 is another example to identify which is the gene influence in the maximum cancer. What not cancer? If you take the breast cancer, if you take the colon cancer, if you take the any liver cancer or something around, how the TP53 is influencing tumor suppressor protein. Okay, and another one is that there is a drug discovery, especially drug discovery. This is a very booming now, especially how you are going to be identify the target, how you are going to be identify the compound, and optimizing that. Just we explained about the molecular dynamic simulations, how we are going to be implement in the clinical trials, especially if you optimize, if you optimize, there is a target and leads, and especially for interactions between the protein and ligand, then we can implement by ADME or pharmacogenomic properties, especially the patient based. Of course, the modern bioinformatics is not just in the biologist who codes, but the data scientist specialized in the language of life. So that means to say that nowadays biologist it's not required a programming language. Why? Why? Because the AI is taking care of now. I'm not bothered about that what code I'm writing now. Because of AI is taking care by prompting. How many people? I think so. Most of the people are aware about that. There is a ChatGPT, Gemini, Growth, and a lot of applications are available, especially large language models. So the question is, sir, I want to learn the large language model. I want to develop a large language model. No, my question is how you are going to be reduce the time of errors by utilizing the large language models. That is playing a major role as being a biologist. And sir, I want to go for the coding. I told you, sir, we have a lot of programming. Like, sir, some people used to go for the Java, some people used to go for the MySQL, some people used to go for a lot of applications and all. Or which programming language is useful, especially for biologists? So my question is, Python is the best, and R is the best source for. If you want to learn, there is a lot of things and all, but the Python and R is open source, which is contains a lot of languages, lot of libraries, and still it is having the development status. And there's one more point, one more guy is the operating system. So I'm working with the Windows, but if the people want to learn the Linux, so how you're going to be implement this one criteria? See, Linux is a source for to understand the large language, understand that there is a large data, understand the big data. The Windows can handle certain level, having certain issues, certain there is a limitations in this criteria. But Linux can change the programming with respect to. We can go for implementing addition to or integrated with the AI. Yes, your job is very good. So the flows, there is a lot of workloads. Industry is required, not from the scratch level. How you are going to implement, especially the flow. So the industry is require the batch processing, not from the there is a struggle activity too. So every time I want to write, there is a automation is playing a major role. If you implement, if you understand, especially the flow based, yes. And if you want to be learn the flow or something around, yes, there is a lot of forums are available, especially the Python based or R based. Yeah. And especially I told you that industry, what industry is looking from the student, looking from the there is a freshers, even the expertise, like, so how you're going to be acquiring the data and how you're going to be understanding the data. So I hope that the most of the people are, especially the sample analysis, sample collection, and sample processing, especially the in vitro, are very good at. I hope. But when you come into the bioinformatics, especially, sir, I required the coding, I required something around NGS, I required some programming, especially that the Python and R. Nowadays, it's that what do you call that era? It is open now, especially the biologists. Without biologists, the programming cannot understand what is the life of the biology to the computer programmer. Let's talk about that one also. So if you want to go for the QC, I told you the quality it is important, validation is important. And if you want to go for the code analysis, especially with alignment with the references or variant calling with comparing with there is SNP single nucleotide polymorphism to check with the clustering between either, for example, I want to, if you take one example like, so the person is affected with the cancer, the liver cancer. See, I don't want the cells from kidney. I don't want cells from the head. I don't want cells from the other than source of the liver. But I want to be implement only the liver, especially the what lobe liver having. There is a right lobe and left lobe. The which lobe is having the cancer. I can target, especially the treatment for target-based therapy. So that is a boom in the especially target-based treatment is available for there is a precision medicine based. Fine. And so this is another agenda like there is a AI discovery. Of course, AI discovery, especially in the precision medicine, and you should be understand there is a multi-omics. The multi-omics are genomics, proteomics, metabolomics, how you're going to express your genes. That is playing a major role here. And what actually there is a biologist thinking. Sir, informatics, it is like a computer programming, and we have there is a computer engineers, their subject. And what are the computer programmers will understand? So that is a biology subject, and biology will be read. And there is another guy, the third guy, it is like there is a mathematics program. So both all together, like the engineering and biologists, both of this is the guy is the mathematician people, mathematician guy. We cannot be understand biology, but bioinformatics is playing a major role of the three subjects of the engineering, biologist, and especially the mathematics. So there is a two different activity like how the builder, how the thinker. See, there is a computational biologist, especially if you have a biology integrated as biology source, you are the computational biologist, you are a thinker. And what bioinformatics engineering can do? The engineering can do that, how develop a program. So developing a script, developing database, and developing it, there is an interface between biology and interface between there is a programming. Okay, and there is another guide if you want to go for the flow based, especially I told you. So there is a engineering person will think about, especially the biologists will think about, bioinformatics engineers think about that, how a pipeline can be batch processing, which integrated with your biology. That is important, playing a role. Without biology, and there is no bioinformatics. And without computer, there is no bioinformatics. If it doesn't have any mathematics, yes, doesn't having any bioinformatics. But integrating with all together, that is very playing a major role. And what computational biology will think about? I told you that the context is the king. Data is the king. Data is the king. The biology, especially the biological data, is the king of biologists, the king of bioinformatics. How you are going to be experimental design? So I want to be there is a add some other liquids, some other solutions, or if you want to go with the PCR, if it doesn't understand the bioinformatics, how the people are doing the PCR, how you are going to be implement the programming language, especially the identification of single nucleotide polymorphism. Yeah, and there is statistical rigor. This is statistical, sir. Statistics means, again, sir, due to the mathematics only, I jump out of there is a BPC, I take in the biology, but again you are telling the statistics. No, statistics what required. If you know the base statistics like mean, median, mode, standard deviation, ANOVA, chi-square, this is the more than enough to understanding as a being artist. And what statistician will be do the latest stage, that is next stage. So one criteria, how you're going to interpret. See, if I'm asking a question like how significantly we are comparing with the one sequence to another sequence, the significant role is a statistician. If you're not understanding what is the significant of this also, I mean to say the significant difference is between the patient and normal. That is a playing a major role. Fine. And if you go for there is essential tech people, like there is a if you go for there is a bioinformatics, especially engineering people, they will write the flowcharts, they will conceptualize your biology, and the making availability in the there is a cloud sources. The cloud sources, we have a lot of cloud sources. See, if you want to go for the commercial based, or if you want to go for there is a free base, the lot of cloud is available. Okay, makes sense. And integration. This is the next guy, the future guy. If you understand, sir, AI means, sir, no, it is not my subject, sir, it's a biology. It's not a biology. It's a totally is a engineering. It's a totally is a no, engineering person. If you ask the question, sir, no, sir, it is totally the statistics. I don't want to go for. The basic of understanding AI is it's not like a chat GPT while writing the prompting something around. No. How the AI can be integrate large language model. For example, I have a whole genome sequencing. I have a whole genome sequence, or I have a whole transcriptic sequence. How you are going to be implement the small applications? How you are going to implement like there is a upregulated genes and downregulated genes? How you're going to be the specific genes, or there is a disease guy is influencing the metabolic pathway. That is important in the air. So that's what. So AI also required. We know very well about the chat GPT, but very limited people know about that bio GPT. You are the biologist, you are giving information to the GPT, and GPT will be storing your data. That is a large language model. And what information you are giving, what information is giving, especially the biology based, then large language model also can be understand. And there is a, if you want to go for the Python is good source, the open source, it's an open learning platform. A lot of platforms are available if you want to learn are either Python and R. So my question is like this, sir, which one preference? So I can't tell that my left eye and right eye both are in the same platform. Makes sense? Yeah. And another one is a translator. So translator is the most valuable hire is the one who can explain the false discovery rate to the biologist and the PCR bias to the software engineer. That is a playing the biologist playing a major role. So the false discovery rate, you know very well about that. If you conduct any experiment, the false discovery rate always will like minimal, limited. So this one you can explain to the biologist means cannot be understand, but should be understand what is the minor limitations is available in the fast discovery rate. So if you go for the mathematician, I can, if I'm asking the PCR means, so what is the method you use to PCR? Is what is the statistical method they asked? Okay, so how you are going to integrating with the three different platforms, that is a playing a major role of the bioinformatician. And there is another one is that there is a data hygiene and governance. This is the next criteria, but very limited activity. If you want to go for the principles, especially the compliance in the aspect of, for example, if you take the what IA is required and what ICMR is required and what CSR is required, especially that if you are identified any drug molecule, if you are identified any new drug, any new protein or novel enzyme, novel protein or novel organism, how you're going to be integrated with S. This is the novel one. How you're going to be writing a report with respect to the supporting document. That is the traceability. And what about the hybrid between the biologists, especially like there's a most of the 90, so 80 people, 80% of the people is handling there is a light skills. But please be behind that. Please be learn some programming language. So the programming, it is next era for especially for the biologist. At the same time, and the data literacy and the value added important, especially for the programming based. Yeah. And so if you want to go for hiring strategy, what people is looking, especially from the biology, especially from the biologist. So if you want to go for the infrastructure, if you want to go for the infrastructure, please be learn that there is AWS, means Amazon Web Services based, or if you want to go for the Linux or something around. So those people are called as a bioinformatics engineer. Sir, no, sir, I want to go for the pipeline based development. You can go for the next floor. So those people are considered as a bioinformatics engineer itself. Sir, I can use the computer application. I can use there is a program language, especially biology, especially microbiology, biotechnology, or whatever biology. I can utilize this. There is a languages, especially for the libraries from the various sources of the R or Python. Yes, those people will consider as a computational biologist. So the new topic, there is a very fancy word. There is AI and BioGPT, and there is a Python. If you want to go for yes, the AI ML is the right now the boom topic, the boom uh, for especially the biologist. Yeah, and no, I don't want to go for the data hygiene. I want to go for the data hygiene. This all program will be understanding the meta gene or there is a principle based, especially all roles. Yeah, I hope you maybe understand what biologist is doing and what bioinformatics is doing. But I'm very sure that I don't want to say myself as a bioinformatician. I want to say myself as a biologist. Even though programming languages may be having the more knowledge compared to the, I want to learn the biology, especially from the you guys. Yeah, I hope you might be enjoyed this today's session and you may understand what biologist is doing, what there is a computational person is doing, as the engineer is doing, what mathematician is doing. So how you are going to be implement in your strategy. And so don't be jump over to the bioinformatician. I'm very clear to you here. Don't jump to the bioinformatician. Being a microbiologist, how you're going to be understand the bioinformatics. Being a biotechnist, how you're going to understand the bioinformatics. Being a pathologist, how you're going to be understanding the bioinformatics. Being a statistician, same criteria too. Yeah, I hope you may understand about the strategies and what industries is looking, especially for the freshers. Thank you guys. Thank you very much.
Wow, that was absolute gold. Thank you so much, Dr. Munikumar, for showing us the big picture and sharing the practical career tips and strategies with our audiences today. Especially the insights on the case studies, they were extremely informative, and you showed us what kind of research is going on, what all emerging trends are there, and actually where the industry is headed to. So overall, the session was truly incredible. I personally enjoyed it a lot. Can we have a huge round of applause in the chat with some clap emojis for Dr. Mani? Yes, and we are going to have the question answer session at the end. So just to remind you all once again about our surprise gift today to be disclosed at the end of the webinar. And in case you're having any doubts, yes, we have answers from our esteemed speakers today. So whether it's about your specific degree or some specific software to learn first, you can just ask away, and we will take up your questions at the end. Thank you once again, Dr. Munikumar. We will have you once again at the end for Q&A session.
Yeah.
I invite to the session now, Dr. Neeraj. I request you to kindly take over the session, sir.
Yes, hello, ma'am. Am I audible?
Yes, sir, you're audible.
Okay, so let me just share my screen, ma'am. My slides are visible, ma'am.
Not yet, sir. Yes, now it's visible, sir. Thank
Okay,
you.
okay, thank you. So, welcome everyone, and thank you for a very detailed and very deep, insightful session to Dr. Muni Kumar. I personally enjoyed this session, and he has almost covered the each and every point which I am going to talk about. So I will just brief you guys about the industry, the internships, the projects, and the job pathways. Okay, so as Dr. Muni have just given you the deeper insights already, so I will be just emphasizing over that how those things will be working for you and what should be your strategy, right? So I will be talking about the strategic career planning and
So I request everyone to kindly be online. I I guess there's some
the strong
technical issue.
foundations that would let you progress towards the more advanced competencies, right? So early projects as well as experience, you have the skills for a specific field, like we are talking about bioinformatics and computational biology. So if you can access the databases, okay, you can understand the databases, you have the programming skills, okay, you can build the models, you can understand the models, you can understand the data sets, okay. So those skills would help you to accelerate or expedite your hiring process, and would bring you opportunities for research collaborations, and they would provide you or guide you towards the graduate admissions, whether they are MSc or post-graduation level admissions or PhD level admissions, right? So, what is the core bioinformatics literacy? Basically, what should you know about the bioinformatics? So, the basics of bioinformatics include the genomics and omics datasets. So basically, the in genomics and multiomics data sets, the sequence formats, that in what form of sequences will be available, and what would be their formats, how you can access those data sets, how you can process those data sets. Okay, so they generally comes with the faster sequence, fast q sequence, BAM and SAM, and VCF files, which are non-negotiable for handling raw sequence data sets. Okay, so these sequence formats provide a strong command over the data sets. So they would present the data set in a defined format. So you just need to just access those data set and you just need to process those data sets for the extraction of what you say, the informational part, right? Then, while you are dealing with the gene expression dataset, so the processing RNA sequence data to quantify expression levels and identify differential expressions. So when you are dealing with the gene expression datasets, so why why you need gene expression datasets? To understand that what are the expression levels, and which genes are expressing differentially. So they might be the point of interest, they might give you the idea about the disease, or they might be the source of disease progression or disease occurrence. Okay, so these are the file formats you generally needs to just work with the bioinformatics, and you work with the gene expression analysis. Then you can also work with the variant analysis. Okay, so calling and filtering SNPs, the single nucleotide polymorphs, and the indels, insertions and deletions, using industry standard pipelines. So GATK is the example of industry standard pipeline. So pipeline is a software, it is a step by step process uh, which have a standard protocol, standard procedure, with which you can just access the dataset, and you can analyze the dataset to find out the SNPs. Okay, or insertion deletions in the given dataset. That would be a gene expression dataset, that would be a genomic dataset, that could be any multi-omics dataset, to find out where are the single nucleotide polymorphs or insertions or deletions that would give you the information, right? So these skills are very basic. If you want to enter into bioinformatics, then you must know that what exactly FASTA file carries. Okay, what exactly FASTQ carries, or what BAM and SAM formats are, why we need VCF, right? And if you want to understand the molecular pathways, then you need network interpretation. So contextualizing data within the biological network using CAG and reactome database. So now you must be aware about these databases, CAG and the reactome. Okay, so it is the Encyclopedia of Gene and Genomes, Kyoto Encyclopedia of Genes and Genomes. You guys can just go to Google after this lecture, just type the CAG and try to understand what exactly it carries. Okay, so these databases are generally carried out to elucidate the molecular pathways, how a particular molecular process is flowing. Okay, what are the interaction points, what are the intersection points, how or where two pathways are getting interacted or intersecting each other, right? So to understand the biology, molecular biology, genetics, genomics, that along with the cell biology and the structural biology, so the amalgamation of all these fields is the bioinformatics. So to understand the bioinformatics, you must need to understand all these terms before entering into bioinformatics. So, what are the high impact technologies? So, computer aided drug designing, which is the computer aided drug discovery. Okay, so it generally deals with the CAD has become a core component of modern therapeutics development because it allows the researchers to evaluate thousands of molecules computationally before performing any wet lab experiments. Okay, so streamline the virtual screening and pipeline development. So the virtual screening and pipeline development, as I just described, that pipeline is a step-by-step protocol to address a specific research question. Okay, so the virtual screening pipeline development comes under the computer aided drug designing because here computers are helping to discover new drug components, okay, or new drug or potential drug candidates. Okay, so if you wanted to just enter into bioinformatics, basically structural bioinformatics, as Dr. Munikumar emphasized over structural bioinformatics, right? You need to understand, I do not say that you must be the expert in these fields, but to start with, to enter into these fields, you must be aware of the molecular docking, as he explained. So the autodoc and autodoc vena are the tools to perform molecular docking. Then the structural analysis, how to visualize, how to analyze, how to just get deeper insights about the structure. You must know the pymol. Okay, They are the critical and modern pharmaceutical research driven tools. Okay, so this is the one technology which is high impact technology. The other one is AI ML integration. So Dr. Muni Kumar has already emphasized there. So I just already said that I will be briefing you about all these things. Okay, sorry. So AI ML integration is, it is the future. The future is data driven. As we know that the data sets are arising, or the data is generating in a humongous quantity. Okay, so we must have the tools, technologies, and the skill sets to address or to understand those data sets, process that data sets, and to extract informational part from that data set, because the raw data is humongous. We need a very small chunk out of that raw data that can represent the informational part, which can provide us the deeper insight, so we can make our decisions. Okay, that is where the AI ML comes in action. Okay, so machine learning models like TensorFlow and the PyTorch, they predict the protein structures. So this is the example of that how AI/ML is providing the, or getting applied into drug discovery. Okay, so AlphaFold is being used to predict protein structures. Even AI/ML pipelines can be used to classify variants and uncover hidden patterns in the massive biological data sets, because they can understand the data set, they can analyze the patterns, and then they can make the decisions on the basis of their learning. That is why they are very high impact technologies. Okay, so, what should be your learning roadmap while you wanted to enter into this field? Okay, so the learning roadmap, you must have the foundation. Okay, so the journey begins with the core biological concepts, basically the central dogma, that how DNA, RNA, and protein, the process flows. Okay, and you must have the basic proficiency about Linux commands. Okay, because you are going to handle the humongous amount of data sets. Therefore you cannot just work with the Windows, so you must have the Linux proficiency. Then you must know the scripting and the logics. Okay, so scripting means you must know that how to write a script, not the YouTube script or the movie script, the programming script that would be handling those data sets, to process those data sets. Okay, so you can understand by this example that you got 1,000 files. Okay, and in those 1,000 files you just need to find out that any particular, so for example those are the genomic files, and you want to see that any particular mutation is there, for example A. Okay, so A is missing or not. So you just write a program, you just run that particular program over 1,000 files, then it would be helpful. The job will be done in just a fraction of seconds. Otherwise, if you just go to address that question manually, that would be very difficult. Okay, so scripting and logics. Then you must know the pipelines and analytics. So you must know that how to build the NGS pipelines, and mastering the data visualization tools like ggplot2 and tableau. Okay, so the pipeline is step by step protocol. Okay, so you must know that how to develop those things. Okay, I'm not saying that you must be expert, but this would be your roadmap. You must have foundation, then you know scripting, and then you can move into pipeline development. Okay, then you must acquire advanced skills, so advanced integration. So you can integrate these skills like cloud computing, AI/ML modeling, and the computer-aided drug designing workflows. Okay, that would be your learning roadmap. Okay, so how would you acquire those particular skill sets, or those um, you can follow that particular roadmap. So you can just go for internships or projects. For example, the first place where you can go, so the academic and national labs like EMBL, EBI, NCBI, NIH. Then in India, they are the CSIR laboratories. Okay, then you can join the summer projects or research internships. Okay, If you want to go into industry focused portals, not in academic or national labs, then you can go to Biotechnica, the R place, where we are also providing the internships and research project internships, the summer projects also, summer and winter projects. Okay. Then nature careers also, the LinkedIn groups also. From there you can just grab or try to just grab a particular opportunity, right? And you can go to the training provider competitions, like the structured intern programs or Kaggle style challenges for biology. And you can also go to the startups or small companies. Even they are providing research interns because they also need workforce, and they do not carry a such amount of very large amount of funds to just cater for the particular workforces. So they just pay a small amount of money, and they just hire interns. So you would also get money as well as the research experience from there. Okay. So you can just choose these options for internships and projects. Okay. Then what would be the student project ideas? Okay, so when you are going to join a particular project or internship, then what should you look for? Okay, you can go for protein modeling. This is basically the structural bioinformatics workflows. Okay, so if you are interested in structural bioinformatics, then you can choose the protein modeling, etc. Okay, where you would get to learn about homology modeling, the structural validation, the short MD, the molecular dynamics refinement, or AlphaFold analysis. Okay, so if you are not interested in bioinformatics but you wanted to learn about multiomics or NGS, that is the next generation sequencing workflows, then you could learn, or you would choose the NGS workflows. So where you would get to learn the design of reproducible pipelines, the quality control, then the alignment, then the quantification, differential gene expression analysis, etc. Okay. Then you can also join the internships with the small data machine learning. So if you want to acquire some advanced skills, then you would choose the ML Pro internship projects, which would be small data, because basically the machine learning or AI would be implemented over larger data sets, but you can start from basics and then you can move to advanced in machine learning. Okay, where you would get to learn the build classifiers, the phenotype prediction using selected public databases, etc. Okay. But if you are a professional, so those ideas for just for the students who are at their graduate or post graduation level, or they are working in PhDs, but who are working in industry. Okay, so who wants to make transition from wet lab or to dry lab? So if you are working in wet lab, whether you are working in cell biology department, biochemistry, molecular biology, any, and you are not interested anymore in that particular domain, then you can also switch to the dry lab. So what would be your approach? So the first approach you can follow, that you can keep your experimental skills with yourself. You can add the scripting skills. You would learn the programming, whether it is Python or any the machine learning programmings. Okay, or the data analytic part, basically the statistics part. Okay, so these are the programming skills. Python are the basic Linux and the pipeline building. The second path you can follow, the complete transition. So you there you can learn about the statistics or machine learning in depth, that would contribute to publicly, and you can contribute those learnings to the public repositories like GitHub or the Kaggle, etc. And you can publish those computational analysis over those repositories. So that would show, or that would demonstrate that you have the skills, and now you are ready to transit into dry lab. Okay. Then I want to just provide you a bonus, that propose joint wet dry projects. They work in a more fashionable way, and they are more productive and document workflows and assumptions. Always just document all your findings, all your assumptions, and what steps you follow, what particular approaches you took while developing a particular pipeline. Okay. So, what job roles you can look for in bioinformatics? Dr. Muni Kumar has already emphasized over computational biologists who are going to build the biological models, who is going to work closely with the experimental teams. Then you could also go for a bioinformatics scientist who is basically going to develop and maintain the pipelines and a large scale data processing and tool development, etc. Then you could be completely a data scientist for life sciences, who is going to focus on the ML applications, that is the machine learning applications and analytics, who is going to analyze that data set using the machine learning pipelines or tools. Okay. And who is going to translate life science questions into data problems, who is going to answer the life science questions with the data sets, and who is going to make inferences just by looking at the data, extracting the information from raw data to the insightful informations. Okay. So what could be your next steps in your career move? So, for students, you can pick any starter project. You can develop your own GitHub repository and apply for internships. For professionals, you need to identify one transferable projects that could be transfer yourself into from wet lab to dry lab, or you can just amalgamate those sequences, but that transferable project must be documented, and you must reach out to mentor. Okay. Where you can just combine dry lab and wet lab sequences, or the wet lab experiments, and you just need to approach a particular mentor who can just mentor you and build your skill sets. Okay. And for everyone who want to just take on the skill sets and want to excel in your career, so you must join our organization, that is Biotechnica, for the talent pool or mailing list, and attend the next onboarding webinar where you would have the better insights, and we will try our best to give you the better insights in the industry or the academics, and that would shape your career, we guess. With this, I would like to thank you, and thank you for providing me the opportunity. So thank you, Urmimala ma'am.
Thank you very much, Dr. Neeraj, for giving us the very practical learning roadmap, very achievable, I must say. And I love the part about core bioinformatics literacy, especially the transition that we need to make from wet lab to dry lab. And the way you have effortlessly explained about the real entry points, it was really helpful. I know so many people struggle with that shift, and you have made it look so easy and achievable. So, thank you once again for your session. So basically, if we have one thing to take away from today's webinar, then this can be the summary: right skills, real projects, and relevant portfolios. So these are your three pillars. Remember that. Webinars like this aren't just for information, it should be for realization, right? So you realized today that reading a textbook isn't enough, and getting the knowledge is just not enough. What the industry demands is hands-on experience with Python, R, Linux, NGS data, as both of our incredible speakers have mentioned. So if you want that career growth, you cannot stay on the sidelines. You have to get your hands dirty with data, right? So I see a lot of questions of you are all asking in the chat that how do I start, or where do I get these projects? So if you want to move from just learning theory to actually building industry ready projects, then I have the answer for you. We are launching the Biotechnica Bioinformatics Winter Internship course starting on 1st December 2025. So this wouldn't be just a class for you. It can be a game changer. It has guided project mentorship. It teaches you all the required tools and skill set. So this is your chance to build that essential portfolio Dr. Neeraj was talking about, and that too under expert supervision. So let this be your brand new year resolution. Do not let another year go by just wishing that you had the skills. So do something, join this Biotechnica's internship, and let's build your career together. So the link is also will be available in the chat right now. It's already there. So thank you, Bopana, for that. And now for the moment you have all been waiting for. First, your surprise, as promised, for staying with us, we are gifting you an exclusive bioinformatics career ebook. It has 20 free bioinformatics tools for you to learn, and you will find the download link in the chat section right now. It's already there. So go and hit the link quickly before it goes away, and it covers a lot of what we discussed today and more, right? And second, yes, let's jump into some questions. Our speakers are here and chat box is open. So let's pick up one question from the audience. If you have any further questions. So Salil has a question. Sir, could you suggest a roadmap for people learning to wanting to learn bioinformatics from scratch? So I think, Salil, we have spoken enough about this, right? Dr. Neeraj has already showed you the flow charts of how you can start. Maybe this question was posted before his session. I hope your doubt is clarified now. Yes, if you could quickly show a thumbs up, right? So let's take Kunal's question. So I request Dr. Munikumar to kindly throw some light on this. So can you show us the kinds of examples of data on which one can work as a bioinformatician? I know you have already spoken about this in the session, but just one-liner summary, I think, for the audience who have joined late. Sir, you are on mute.
Perfect. Yeah. See, there's a lot of databases are available, guys. If you want to go for the basics, like NCBA is the best source to understand the basic level. And if you want to go for the advanced, even NGS, especially if you want to go for any machine learning program or artificial intelligence program, the NCBA is having the immense information. And another one is that there is a EMBL, ENE, European Nucleotide Archive. So there is another source to get the secondary data. So the question is, what objective you require? You want to go with, there is a CADD, means computer drug designing, or you want to go for the, there is a NGS analysis, or you want to go for the machine learning. That is a playing a major role. And one of the slide I mentioned the basic to an advanced level, and either the disease level, or even to go for the drug designing level, or even go for the basic understand the database also. I given one of this slide that is useful for the very freshers people like the fresher students. Yeah.
Thank you, sir. So yes, fresher related questions. We had a lot of questions. So I think you all are listening to Dr. Muni Kumar, the advice that sir has given. Okay, so now we would like to take up a question from Yatin. MK, for getting a good job in this field, do I need to do an MSc and PhD, or is doing internships enough? Dr. Neeraj, if you could guide on this, please.
Yes, ma'am. Thank you for your question. So, if you want to, this is a very debatable question. However, if you want to get deeper insights about a particular topic or if you want to specialize one skill, then you must go for MSc along with the internships. But if you want to just understand that how things are working, if you want to specialize your skill set and broaden your skill set, and you are a research driven person, then PhD is the most suitable option for you. Okay, if you're just looking for a job, then MSc plus your internships is enough for your job, of course.
Rightly said, sir. Even I agree with that. Thank you for answering. Dr. Muni Kumar, we have an interesting question here. If you could throw some light on this, what is the industry versus academia situation in bioinformatics in India right now?
Who's the guy that asked the question? It's a fantastic question. See, industries, I'll be explaining that what required actually. Correct. So, see, if you want to grow in the bioinformatics, I'm telling you guys, I will give you one small suggestions to you. Take any one programming language. Please be perfect on that, the basics to the advanced level. You are the pro in the mathematics. Sir, you may ask the question like, sir, Python is available, R is available, Java is available. Which one I have to go for? Anything you should be check with the either the Python means you should be stick on to the Python. Either you want to go for the R, you can stick on to the R. That is industry is required. But what about the academia? Academia is required. There is a one understanding like the project handling. The project handling like there is, for example, if you go for there is NGS with respect to the AI. Whatever one programming language is enough. If you take either R or Python, anything you can take one, and the objective will fulfill. Objective will fulfill based on the scripting or anything. And another one is that especially in the academia, there is a publications, especially the presentations, especially there is international conference and national conferences to attend something around. So that academia is required. So the majorly I used to be encourage the students, especially and see, report writing is a playing a major role, and paper writing is a playing a major role in academia. But industry, as Niris mentioned about that, make it like your portfolio in the GitHub and make it like the programming language, especially that there is a lot of like Niris has mentioned the Kaggle. The Kaggle is a very open source for it is the replica of your system. Yeah, you can write the a number of times in the program and all. Yeah, I hope I may be clear about the question itself.
He has a follow-up question, Dr. Munikumar. Could you, he's asking, yeah, is there a substantial pay gap? PhD versus MSc for industry and bioinformatics. That's an extension of his question.
See, MSc, that's what, just to the extension of the same answers to me. See, uh, the PhD and MSc is the only degree difference, guys. There is no difference at all. The MSc might be having the more academic, and the PhD might be having there is a one pinpoint objective. But make sure, being MSc or being a PhD, what objective you want to learn. That is required in the academic and both in the industry also. The objectives are playing a major role, and the object also should be fulfilled. It should be fix it out. It should be fix it out. Either I want to be grow in the air, either I want to go in there is a NGS. That is the playing a major. Yeah.
Thank you so much, sir. So, Dr. Neeraj, there's a question after biotechnology, what to do, sir? Which field is best option for higher education? And it's not a follow-up question, but then we have one more question uh, from Rosa in LinkedIn that I'm a biotechnology engineer and I would like to dive into bioinformatics world. Do you suggest to start with a master? So basically, some career advices they want. If you could give a combined answer for options after graduation, that would be really helpful.
Sure. Thank you for your questions. First of all, both of you. So the question itself is a very what do you say, a very curious question. I think it is because after graduation you have a number of options. Okay, but I basically as per my experience I suggest yes, bioinformatics is a very good option to choose as per current scenarios. Okay, because here you get to learn a number of things. You can integrate knowledge from a diverse disciplines. However, you need a very specific skill set also. Are you ready to build that particular skill set or not? Okay, so programming is not a must-have skill, but it is a very crucial and necessary skill. Okay, and most of the people struggle with the programming. Okay, so because in these days everyone knows that ChatGPT or the Gemini, any AI assistant can write programs for yourself. But do you understand that program? Does that program is serving for you or you the task you wanted to achieve? So for that you must have the knowledge. Okay, so assistance would help you. Okay, so now again the question come to that particular point. It is completely up to your choices. So if you are ready to shift into bioinformatics and you have the courage to learn new things, endeavor into new skill sets, then it is completely yes from my side to switch into bioinformatics. But if you are not ready to just broaden your skills or to embrace new skills, then I think you must think before jumping into bioinformatics. But bioinformatics is have a very bright future, and in coming days, because the data is rising at a humongous rate, so bioinformatics is a very brighter field from my side. Thank you.
Right, sir. Thank you, Neeraj sir. So we should make informed choices. You have the information, now you need to think it through, as sir rightly said. So we can take up just one last question. If you have any further questions, you can drop it in our community groups. Yes, so Dr. Muni Kumar, if you could please guide on this one. Python or R language, which coding language should one prefer? And can a person with no coding knowledge learn this easily? And here's an extension question also. Should we need a certification in R or Python for getting a job in informatics?
Oh, I think so. Yeah, I think so. Neeraj is answered, I think so, this question actually. See, even myself also, I'm telling you, see, be prepared for that one programming. Which one? So you may ask the question, sir, Python having the five letters and R is having one letter. Can I select the R? In that point of view, I will say no. I will say no in that point of view here. But as Neeraj has mentioned, we have a lot of models like large language models. How you are going to implement? See, in if you take there is a there is 3 years back and 4 years back something around. The people used to discuss about in the forums. So, for example, you take the R contain the R forum to discuss whether there is expertise in R to writing any discussion between any issues related to the errors and all. But nowadays, it's not required in the forum itself. You can use the GPT, you can use the Gemini. There's a lot of explanation related to. But the point of understanding, either sometimes GPT might be facing the issues like sometimes facing giving the information is wrong, but you are the decision maker. The decision has to be that is a correctly or not. That is a playing a major role. But in this point of view, R and Python, the both are in the same way. Both are in the same platform. The which platform you want. Yeah.
Right, sir. Thank you so much. Okay, so unfortunately, looking at the time, we have to wrap up the Q&A here. Many are asking that how do you join these internships and our Biotechnica projects like internships? Yes. So we do have projects guided by your very own principal investigators, project investigators. They are going to guide you at every step, and they are certified project internships for various durations like 3, 6, and 12 months, along with the bioinformatics course. So the link of this, how to join this, you just have to click on the link which our team is posting in the chat box. Okay, so just check your chat box and your surprise free gift from us today. That is an ebook on 20 bioinformatics tools. You can also click on that link. So both the links have been posted once again. Kindly check that out, right? So to those questions we couldn't get to, don't worry. Our team will try to reach out. You can just drop them in our community groups. So before we end, I want to leave you with one final thought. We covered a lot today, but here's the honest truth. Information alone does not change your career. What does? Action. You have to take action, right? So please don't let today be just another webinar you watched and forgot about by tomorrow morning. Don't let this excitement fade away. You have the roadmap and you know the destination. Now you just need to take the first step. Okay. So if you want us to hold your hand throughout your journey, the Bioinformatics Winter Internship starts on December 1st. The link is pinned in the chat and it's the bridge between knowing what to do and actually doing it. Okay. And a massive thank you to all our incredible speakers. Dr. Muni Kumar, thank you once again for taking time out of your busy schedule and sharing your wisdom with us today. And thank you so much, Dr. Neeraj, for enlightening us on very important aspects. And also a huge thank you to you, amazing audience, for your time, your energy, and your brilliant questions. I'm sure many of our queries got clarified today. So, on behalf of the entire Biotechnica team, I want to tell you that the future of biology belongs to those who can decode it. Go out there and start decoding, right? So, good night, everyone. Stay curious, and we will see you in the internship and in our next webinar. Bye.